The overall goal of this impression cytology technique is to derive enough cells to immunologically phenotype the cells involved in ocular surface disease.
This method can help answer questions in the field of ocular surface, such as which immune cells are important for ocular surface inflammation. The main advantages of this technique are that it is relatively non-invasive and simple to perform in the clinic and the lab.
The implications of this technique extend towards the therapy of ocular surface inflammatory disorders, since it allows the identification of specific populations of immune cells.
This method can provide insight into the ocular surface immune cell population. It can also be applied to the investigation of ocular toxicology and ocular fibrosis.
Generally, individuals new to this method will struggle because of a lack of experience in handling the low number of cells collected from the eye.
After determining the participant's clinical status, apply 1 to 2 drops of topical anesthesia to the superior bulbar conjunctiva and the inferior fornix of the participant's eyes. Wait 4 to 5 minutes for the stinging sensation of the anesthetic to wear off. Then position an impression cytology device tangentially onto the conjunctiva on the temporal bulbar side and gently press and hold the push button for 2 to 3 seconds. Using forceps, release the bulk harvested ocular membrane into a microcentrifuge tube containing 1 milliliter of culture medium, followed by 1 minute of